STB1093 Group 3 Subgroup 3 Practical 4.
Gram staining is a staining technique used to classify a bacterial species into either the gram-positive or gram-negative group. Gram-positive bacteria retain the initial crystal violet stain, purple, whereas gram-negative bacteria are decolorized and stained with carbonyl fusion or safranin that appear pink. The name of the technique is derived from Hans Christian Gram, a Danish bacteriologist who pioneered the technique in 1882 to identify the organisms responsible for causing pneumonia. The initial step is to heat fix the smear from a microorganism. Next, crystal violet dye is used for the first staining. The dye is then fixed with iodine, forming a crystal violet iodine complex that prevents the dye from easily being removed. Next, a decolorizer such as ethanol is used to remove the dye. The final step is to counterstain the smear with safranin. According to Tripathi and Sapra, the ability of the bacterial cell wall to retain the crystal violet dye during the decolorizing step is the working principle behind gram staining. Gram-positive bacteria have a thicker peptidoglycan wall, whereas gram-negative bacteria have a thinner peptidoglycan wall and an outer membrane composed of lipids. The use of ethanol dissolves the lipid outer membrane of a gram-negative bacteria, causing it to lose the crystal violet stain. The purpose of counterstaining is to give the decolorized gram-negative bacteria a pink color for easier identification.
Materials and methods for this experiment involve a few gram stain reagents. Firstly, Harker's crystal violet. Secondly, grams iodine. Thirdly, 95% of ethyl alcohol. Fourthly, safranin. Fifthly, microscope slide. Sixthly, staining rack. And seventhly, bibulous paper. For the methods, first of all, we need to clean our workspace using aseptic techniques. Next, we take a microscope slide and label it. It is important to recognize the glass slide that you put your sample on. Next, add a drop of distilled water on a microscope slide. Use the inoculating loop to take a single colony to transfer to the glass slide and make a thin smear of the bacteria sample on the slide by making a circular motion. Please make sure to do the circular motion until it dries out. Keep fixed specimen by passing the slides through flames a few times. The slide was placed on the staining rack and each smear was dropped with crystal violet. The crystal violet washed off using tap water and excess water is drained off after 1 minute. Each smear was dropped with iodine. The iodine of each slide were washed with tap water after 1 minute and dried out. Each slide tilted and decolorized with 95% of ethanol until colorless appeared and the alcohol drained after 15 seconds. The alcohol was drained with tap water. The smear was counterstained by safranin for 20 to 30 seconds. Each slide washed with tap water and proceed to drying. A tissue paper can help aid the drying process. Lastly, each slide is observed by a microscope until 100 times magnification with the aid of immersion oil. The step was repeated for both slides.
This is the result that has been observed under the microscope. On the left one is sample A and the right one is sample B. For sample A, the Gram reaction is gram positive, and the serum morphology is appeared to be purple in color, rod in shape, and the arrangements is in pair or palisades. For B samples, the Gram reaction is gram negative and appeared to be pink in color and have a spherical rod shape, the intermediate between the two, and the arrangements is in single and clusters. As what observed, sample A possessed purple color stain, meanwhile sample B possessed pink color stain. Sample A is the gram-positive bacteria, meanwhile sample B is a gram-negative bacteria. Also, the bacteria in sample A is pairs and palisades in arrangement, meanwhile sample B is single and clusters in arrangement. Peptidoglycan, a structural component of bacterial cell walls, is the target of the gram staining procedure. According to Madigan et al., the purple color in sample A results from the retentions of the crystal violet stain by gram-positive microorganisms. This occurs due to the thicker peptidoglycan layer in the cell walls, effectively trapping the crystal violet iodine complex. Conversely, the pink color in sample B indicates that gram-negative bacteria did not retain the crystal violet stain, but instead took up the counter stain, typically safranin, that was used in this experiment. The thinner peptidoglycan layer and the presence of an outer membrane in gram-negative bacteria allowed the crystal violet iodine complex to be washed out during the staining process. So, the gram-negative microbes stain as pink due to the decolorization step. This step disrupts the thinner peptidoglycan layer and outer membrane, allowing the crystal violet iodine complex to be washed out, enabling the uptake of the pink counterstain safranin. So the difference between gram-positive and gram-negative is that the presence of outer membrane that will later result in staining of pink color. In this experiment, we use 4 types of dyes. The purpose of each dye in the staining process is as follows. The first one is crystal violet, which is the primary stain that imparts a violet color to all cells. The second one is iodine, act as a mordant, forming a complex with crystal violet and enhancing its retention in gram-positive cells. The third one is the alcohol. Alcohol is used as decolorizer agent that differentiates gram-positive and gram-negative cells based on their ability to retain or lose the crystal violet iodine complex. And the last one is safranin, which is the counterstain that imparts a pink color to gram-negative cells after the decolorization step. Then, we will also have precautions that are essential during the experiment. The first one is caution is needed to not over-decolorize, as it may lead to false results by removing the stain from gram-positive cells. The second one is drawing behind a glass plate is advisable to avoid contamination, ensuring accurate and reliable results. The third one is that sterilization of loops is crucial to prevent contamination, which could result in less accurate findings and errors. And last one is that we need to gently pat and blot the plate rather than wiping it to help maintain the integrity of stain samples. Post-lab questions. The answer for question 1 is without the iodine step, gram-negative bacteria would likely to appear colorless or faint, and gram-positive bacteria would retain their purple or blue color from the crystal violet stain. Question 2. The answer for it is yes. While other dyes like gentian violet or methylene blue can be substituted for crystal violet, and alternatives such as fuchsin can replace safranin in the gram stain procedure, the results may vary and their ability to clearly differentiate between gram-positive and gram-negative bacteria could be compromised. The original combination of crystal violet and safranin is well established and widely used, with the specific staining properties that contribute to their reliability of the gram stain method. The gram stem involves a series of steps, including the application of primary stent, commonly crystalline violet, iodine as mordant, decolorization, and a counterstent, commonly safranin. The choice of dyes affects the abilities to differentiate between gram-positive and gram-negative bacteria. Question 3. In the gram stent procedure, the mordant, the iodine, plays a crucial role in the stenting process. After the application of the primary stent, the mordant is used to form a complex with the dye within the bacterial cells. This complex is essential for the retention of the primary stent colorization step. The formation of this crystal violet iodine complex helps to prevent the removal of dye during the decolorization step. The gram reaction of bacteria is correlated with differences in cell wall composition, where gram-positive bacteria have a thick peptidoglycan layer and retain the crystal violet iodine complex, while gram-negative bacteria have a thinner peptidoglycan layer surrounded by an outer membrane. This distinction influences susceptibility to antibacterial agents, with gram-positive bacteria generally more susceptible to antibiotics targeting the cell wall, while gram-negative bacteria may exhibit greater resistance due to the presence of an outer membrane.